Well, good afternoon, and thank you, Heide, and the organizers for the invitation to speak here today. I know we are trying to give a little time to some of the people who are coming from Dr. Osrai's talk, but I would like to start by highlighting some of the, sorry, I need to get my pointer to work. Okay, here it is. So I have no financial disclosures to talk about. And for this audience, it's not news that breast cancer brain metastasis is a significant problem with between 15 and 20% of breast cancer patients with metastatic disease developing symptomatic brain metastasis. Now, in addition to the therapies that patients receive for the treatment of their primary tumors or systemic disease, brain metastasis often includes the addition of radiation in the form of stereotactic radiosurgery or whole brain radiotherapy, which is now a bit less used, and also surgery. And all of those therapies alone and in combination have significant motor and cognitive consequences for patients, in addition to the obvious damage of brain metastasis growing in an organ that has no room to go. And we know that some of the limitations to the effective treatment of brain metastasis come from multiple issues, part of the inability of many of the drugs to effectively cross and target brain metastasis due to the brain tumor barrier. Also, for a better understood now phenotypic switch that happens between breast cancer cells, between the primary tumor and the brain microenvironment, and also, like I mentioned, the unique toxicities of radiation and other therapies in the brain. And we have always thought about brain metastasis as a disease that progresses particularly in women who are affected by triple-negative breast cancer and HER2-positive disease. And this graph here, however, highlights that there is a significant cumulative incidence of brain metastasis also in luminal breast cancers and estrogen receptor-positive cancers that happen during time. However, like in the extremes of these sort of aggressive subtypes, patients with triple-negative breast cancers can have 30% or 40% of incidence of brain metastasis, and these women tend to be younger, so brain metastasis incidence in this cohort tends to happen in younger premenopausal women, often at the time of diagnosis. By contrast, and you could probably appreciate that in here with this, in this graph, patients with estrogen receptor-positive breast cancer present with brain metastasis at a lower incidence within the subgroup. Only about 10% of patients develop brain metastasis. However, because this is a large proportion of patients with breast cancer, this number is not trivial. Now, these tumors tend to develop in response to estrogen, but patients are treated with endocrine therapy. So brain metastases tend to appear in this patient population in a postmenopausal setting or post-endocrine therapy, which suggests that there could be aging and hormonal changes that are associated with women age and menopause that could be influencing brain metastasis progression and responses to therapy. So we have been asking sort of the questions about how age and hormonal changes that come with age are important for brain metastasis. And we know that young age is an independent predictor factor for the development of brain metastasis, which just means that even for patients with different subtypes, if women are diagnosed when they are premenopausal, they have a higher risk of developing brain metastasis. We always thought that this was a consequence of triple-negative breast cancer being more common in younger women. However, even within triple-negative breast cancer, when women are diagnosed premenopausal, the incidence of metastasis is higher. And we always think that the part of the reason this incidence occurs is that estradiol, the main premenopausal hormone, is able to act through estrogen receptors, which are expressed in many cells within the stroma of brain metastasis. This is just an example here of a tumor that is negative for estrogen receptor, but the stroma is clinically estrogen receptor positive. And this estrogen acts through classical estrogen receptors in these cells, but you also can appreciate here that estrogen receptor within astrocytes and the stroma in the brain can also act through membrane-initiated signaling that ultimately leads to activation of gene transcription and changes in different cells within the microenvironment. So in addition to have estrogen receptors, the brain, and particularly astrocytes, are able to synthesize estradiol as a function of the enzyme aromatase, which catalyzes the conversion of testosterone into estradiol. And these reactive astrocytes, which are associated with many brain pathologies, and they are associated with brain metastasis progression, as you can appreciate here, are present in many brain metastasis patients, which suggests that both brain and ovarian estradiol might be contributing to the progression of brain metastasis. In fact, we have shown through multiple models, both in immunocompetent and immunocompromised mice, that if we overectomize mice and we supplement them with premenopausal levels of estrogen, or we just keep them overectomized, or supplement them with the aromatase inhibitor letrozole, we can see a significant increase in the amount of brain metastases that are formed in mice when they are injected with cancer cells that are themselves not responsive to estrogen. So these MDA231 cells, 41BR cells, and E071 cells, these cells are not responsive to estrogen on their own, but they increase their ability to metastasize to the brain. They form multiple metastases and larger metastases when mice are treated with estrogen as compared to mice that have been estrogen suppressed. Mechanistically, we have demonstrated that reactive astrocytes are very important for mediating this response. As I mentioned, they are abundant in the brain. They are part of the neuroinflammatory response that is important for brain metastasis. And here we are using just an example of an in vitro system in which we use a monolayer of reactive astrocytes to serve as a feeder for our cancer cells that are labeled in green. And what I hope you can appreciate is that these cells don't grow in response to estrogen on their own, but when you culture them with reactive astrocytes, here in black, they grow more. They grow much more when they are co-cultured with estrogen, and you can use an aromatase inhibitor or an selective estrogen receptor modulator, tamoxifen, or an antagonist ICI, and you can block this paracrine effect in proliferation of these cells. We have shown, in the past that mechanistically, estrogen can activate estrogen receptors in this estrogen receptor-positive astrocyte, leading to the upregulation of a number of different growth factors, including BDNF and EGF, which in a paracrine manner can activate our better-known growth factors that drive growth of triple-negative breast cancer, such as EGFR and TRAC-B, or ARCA2. Now, the activation of growth factors in a paracrine manner leads then to increased migration, proliferation, and ultimately more brain metastasis. But importantly, if we block estradiol production using aromatase inhibitors or avarectomy, we can block this colonization, as well as using BDNF inhibitors or EGFR inhibitors for block metastasis. Now, we know that these growth factors are not the only things that are activated metastasis, particularly because astrocytes play a very important role in the management of neuroinflammation in the brain. So what you can see here is estrogen has been well known to be a modulator of neuroinflammatory responses in pathologies. So we have asked to what extent estrogen can modulate immune cells associated with brain metastasis. To ask that, we have done single-cell RNA sequencing of immune cells isolated for the mice that have been treated with these different hormonal conditions. And as you can appreciate here, the presence of estrogen in these mice really changes the subtypes of microglia that are associated with the metastasis, and particularly suppresses microglia that are able to express immunomodulatory markers. So here on the right, you can appreciate that a lot of the antitumoral normal responses of microglia are repressed by estrogen, and they are reactivated when mice are treated with ovarectomy alone or in combination with letrozole, which led us to think that it is possible that use of endocrine therapies could be used in a therapeutic setting to decrease the progression of metastasis and help modulate the microenvironment. In fact, if we look at how those microglia could be impacting other immune cells, this is just to show that not only is changing the microglia programming, but it's also estrogen is repressing the recruitment of immune effector cells like T cells and NKT cells to the brain microenvironment. And if we wanted to test again whether this immunosuppressive role of estrogen could be counteracted by using endocrine therapies like putting the mice in estrogen withdrawal and letrozole. And as I mentioned at the beginning, the standard of care for brain metastasis often includes radiation, and we know radiation is a critical modulator of responses in the brain. So when we use these therapies in combination with radiation, we see a significant decrease in metastatic colonization, progression in these mice using the model of the E071 cells, as well as the 41 model here, showing again that the combination of endocrine therapies with radiation seems to be effective to decrease the progression of metastasis. Now, this is not a cure. These mice still have metastasis and they will eventually succumb to the disease, but this demonstrates that there is a modulatory event of radiation with immune cells within the brain. Now, we know this event is mostly regulated through the modulation of immune cells, because when we perform this experiment in the absence of radiation, we actually don't see an effect of endocrine therapy, indicating that the radiation and probably the antigen presentation effect of radiation is required to potentiate the effect of endocrine therapies. Nor we see this effect when we perform the same experiment with the same cells in an immunocompromised host that lacks anti-NK cells. So I don't have time to show you all the data, but we have evidence that endocrine therapy plus radiation synergize to decrease the progression of estrogen receptor negative brain metastasis through the modulation of the immune system. And just to summarize this part, what we have evidence, and that is, interesting correlative evidence in clinical patients suggests that estrogen, premenopausal estrogen in younger women, could be promoting estrogen receptor negative cells growth through paracrine activation of the growth factors that I mentioned, EGFR and TRKB, which importantly can be targeted with FDA-approved TRK inhibitors. Additionally, we have evidence that estrogen promotes an immunosuppressive brain microenvironment that is contributing to metastatic colonization and progression. And all of these lead us to think that these endocrine therapies might be used in combination with stereotactic radiosurgery or whole brain radiotherapy to decrease brain metastasis progression in younger women. But it also poses a very important question, is that I mentioned at the beginning that patients with estrogen receptor positive breast cancer are the patients with the lowest incidence of brain metastasis as a subgroup. While this fraction of patients is not insignificant, it begs the question that if part of the reason these patients do not progress to brain metastasis as fast is because they are treated with endocrine therapies very early on, that could be contributing to the early control of metastatic colonization in the brain. Unfortunately, there is no way to dissect what endocrine therapies do directly into the tumors of what they do in the microenvironment for those systems. But trying to figure out how is that estrogen receptor positive brain metastases are happening, because when we were doing our studies with triple negative breast cancer, we realized the fraction, and here this is exemplified, of patients with estrogen receptor positive brain metastasis is not small. Most than a half of the patients samples that we have with brain metastasis come with patients that have estrogen receptor positive disease. And if you look here, you can appreciate that the majority of these patients at the time of these samples are either postmenopausal because of age or because they have been in anti-endocrine therapy for a long time. Now, there has been some caveats to studying estrogen receptor positive brain metastasis, and is the fact that as a property of estrogen receptor positive breast cancer, the progression of the metastases are slower. There are no brain trophic cell types that go to the brain that are ER positive. And the majority of breast cancer cells of ER positive breast cancer require estrogen supplementation to grow in the mouse, which immediately puts us in an environment that does not mimic the progression of the metastasis in patients. We have joined forces with Dr. Carlos Sartorius and Peter Cabos at University of Colorado, who developed a panel of estrogen receptor positive PDXs. And when we put these intracranially in a model of experimental metastasis in the mouse, we can see that there are some of these PDXs that have an increased ability to go to the brain. So what we ask here is, can we develop models that mimic the progression of estrogen receptor positive breast cancer? So to do that, we put these cell lines into younger mice or older mice that have been overectomized or not, and we asked to what extent the microenvironment might be driving differently the progression of ER-positive breast cancers. And so I hope you can appreciate here that in younger mice, these cells are fully dependent on estrogen to grow. So if we don't supplement with estrogen, these tumors don't grow, and these are tumors in the brain. But if we do this in age sham, and by age in the NSG mice, I'm talking about mice that are above 50 weeks old, we see a trend to an increased brain metastatic progression in the absence of estrogen supplementation. We went ahead and confirmed that this increase in brain metastasis is not due to the low levels of circulating estrogen in the older mice, because if we overectomize those mice, the incidence of metastasis continues the same. And I hope you can appreciate here that even in these age mice, either sham or OVEX, the metastases are macrometastases that can be even identified via MRI. At the time that we were studying this, there was a seminal paper that described that amplification of FGFR1 was the only genomic change that was associated with an increased risk of late recurrence in postmenopausal women with estrogen receptor-positive breast cancer. And we know from the neuroscience community that FGF2 and FGFR1 signaling is very important for brain function, also is important for promoting endocrine resistance in estrogen receptor-positive breast cancer. And in many subtypes, FGFR1 signaling increases the risk of brain metastasis in metastatic breast cancer. Surprisingly for us, these cell lines, or these PDXs that have the increased incidence of brain metastasis, are exactly those that have amplification, genomic amplification of FGFR1. Now, we know also that this genomic amplification of FGFR1 causes increase in FGFR1 expression, but they are not associated with autophosphorylation of FGFR1 in these samples, which indicates that this FGFR1 is still susceptible to be active through the microenvironment. So we went ahead and tried to ask to what extent FGFR1 is important for the ability of estrogen receptor-positive breast cancer cells to grow in the brain, both in response to estrogen in the younger mice, as well as in the old microenvironment. And I hope you can appreciate here, in both cases, deletion of FGFR1 or downregulation of FGFR1 is sufficient to significantly decrease the ability of these cancer cells to grow in the brain. Now, the knockdown or the downregulation of FGFR1 in these cells in vitro is not full. It's not full deletion completely kills the cells. This is a downregulation, which indicates that FGFR1 is still important for the cells, but the cells don't change dramatically their ability to grow when they are in vitro. So this is somehow an ability of the cells to respond to things in the microenvironment. And so, as I mentioned, FGF2, which is the canonical ligand of FGFR1, is very important in brain function, and it happens to decrease with aging. And this is a quantification of FGF2 expression in primary cells isolated from mice that are young or aged based on the age cutoff that I mentioned. And you can see that there is decrease of FGF2 as well as in FGF2 in astrocytes that had been aged in vitro using a long-term culture. So consistent with this decrease in FGF2, when we co-culture these cancer cells with young astrocytes, we can see that these astrocytes are able to promote estrogen-independent growth to levels that are similar to what estrogen will do in these cells that are highly estrogen-dependent. We can see that FGF2 is able to activate signaling in these FGFR1 cells. The conditioned media of astrocytes can do the same, and the use of an FGFR1 inhibitor is able to block partially this signaling induced by these young astrocytes. So when we culture this with young astrocytes and old astrocytes, we see that the loss of FGF2 in the old astrocytes is associated with a decrease, with an impairment in the ability of these astrocytes to support growth in these cancer cells. And if we knock down FGFR1 in these cells are completely unable to colonize the orthocolonelino to grow in co-cultures with astrocytes in this co-culture system, which indicates again that the loss of FGF2 in the astrocytes might be important for the old brain. But what is important here is that astrocytes are losing FGF2 in the age microenvironment, but if we co-culture these cancer cells in organotypic brain slices of older mice, there is a still growth of these FGFR1 positive cells and dependencies on FGFR1 to grow in that microenvironment. So there has to be mechanisms outside of canonical FGF2 activation of FGFR that are implicated in the growth of these FGFR1 positive cells in the old microenvironment. And so FGF2, as I mentioned earlier, is a very important driver of synaptic plasticity and formation in the brain. FGFR1 has non-canonical binding partners. One of them is NCAM, that regulates the synaptic plasticity, and it does so by bringing close proximity between cells that have FGFR1 and NCAM, and allows the formation of synaptic punta that are important for the uptake of glutamate and other resources in the normal synapses. And the field has recognized that cancer cells now are able to form similar connections with neurons. The reason we thought this was an important avenue of alternative pathway to activate FGFR1 is that FGF2, even though it has much higher activity for NCAM, decreases with age, decreases with estrogen, but NCAM is constantly present on the cell surface of neurons and astrocytes at a very high concentration. So in the absence of lower levels, or in the presence of lower levels of FGF2, NCAM is stoichiometrically much more abundant than FGF2 in the brain. So what we hypothesize is that this NCAM could be driving FGFR1 activation and the formation of cancer neuron synapses in the age microenvironment. Here, I just want to show that if we use a very low concentration of NCAM in vitro, we can see downstream activation of FGFR1 that can be abolished by FGFR1 inhibitors. And if we knock down or this experiment actually is showing that the membrane-bound NCAM in a cell line that is a neuroblastoma, it exemplifies neurons, is able to, in a paracrine manner, activate FGFR1 in cancer cells. If we knock down NCAM in the neuroblastoma cells, now they are unable to induce upregulation of FGFR1 in the cancer cells, again indicating that this proximity of NCAM-positive cells could be a driver of FGFR1 activation in the brain microenvironment, particularly in the older setting. Now, this is important for what I mentioned before, this new idea that cancer cells can form pseudosynapses with neurons, and that by doing so, they are able to take metabolic input from both astrocytes and cancer cells. And so what I hope you can appreciate here is that there is just higher density in this top row compared to the other ones. This density is marking the synaptic punta, which is a colocalization of synapsin and PSD95. And what it basically is showing here is that if we knock down FGFR1, or if we use an FGFR1 inhibitor, we can see a decrease in the number of synaptic puntas, suggesting that there is a function for FGFR1 in the formation of synaptic punta and the growth in the brain. And again, this is important because it has been recognized that the formation of these cancer cells' synaptic interactions are critical for the outgrowth of disseminated cells in the brain. So I hope that I gave you a taste of some new mechanisms that impact the growth of brain metastases in estrogen receptor-positive breast cancer. I think the point that I really wanted to make here is that we do need to consider the host in the modeling of our estrogen receptor-positive breast cancers and model the progression of brain metastases in a setting of endocrine therapy resistance, low estrogen drive proliferation, and aging. We have some evidence that these brain-specific mechanisms, including the functions of FGFR1, could be contributing to the late outgrowth of brain metastases in estrogen receptor-positive breast cancer. And we are finishing some studies to elucidate whether FGFR1 inhibitors have similar effect in the setting of low FGF2 kinase activity of FGFR1 as compared to NCAM-driven activation of FGFR1. And lastly, I think it's an important point to recognize that the late progression of estrogen receptor positive brain metastasis might be resulting from the acquisition of genetic changes that provide growth advantage in this estrogen depleted age microenvironment, probably as a result of therapy responses. And finally, I want to acknowledge the people who did a lot of the work that I show you. The first part of the estrogen story is mostly the work of Maria Jose Contreras and Karen Alvarez in the lab, who are here and here. And the second part of the story is the work of a graduate student, Morgan Fox, and Jenny Jaramillo, and all of the funding forward. It's very important to highlight that this project, particularly our second project, was inspired by Leanne Kramer, who is here in the audience, and supported by patient advocates Christine Hodgdon, who is in the panel. So part of this work is definitely the result of collaborations with them. Thank you.
Is this on? No. Can't turn it on. There we go. Thank you. So that was a great talk. We're going to move right into the next talk, and again, hold the questions until the end. So this next talk is by Peter Vermeulen, who's at the GZA Hospital in Antwerp, Belgium, and is going to talk to us about the biology and treatment implications of organ-specific liver metastasis. With that.
Thank you. Okay, I have nothing to disclose. If we look at breast cancer liver metastasis, well, if you want to define the scope of the problem, about 5% of patients with breast cancer will indeed have metastasis at diagnosis, and about 30% will eventually metastasize, of which 50% will have liver metastasis, so really huge problem. Luckily, but still a lot of work to do, I think, median survival has been shifting from 2 to 3 years to 5 years. But there are some important outstanding clinical questions that we're not going to solve during this session, but an important question is how to optimally treat an individual patient with liver metastatic breast cancer. So, a related question is, of course, for which patients is local treatment of liver metastasis indicated, and do we look only at number of metastases or also at growth kinetics, and probably the latter is very important. And most importantly, can we really develop a treatment that is targeted to the cancer cell in his new environment, which in this case would be the liver microenvironment. So really trying to get from biology to treatment implications. Okay, we all know the metastatic cascade, and this is luckily very inefficient, this metastatic cascade, and probably because a cancer cell encounters sequential different microenvironments that are probably and luckily very aggressive to these cancer cells. So in our lab, the overarching research question, and probably in a lot of your labs, is how does a cancer cell from a mammary gland, and that is used to grow as a mammary tumor, is able to adapt when it arrives in the liver and gives rise to a growing metastasis? I think that is a fundamental question and not an obvious one to answer. And if you look at this adaptation from a morphological point of view, and this will be the core of my talk, is there are essentially two different ways in which a cancer cell can adapt in the liver and can give rise to a growing metastasis. So not all liver metastases are equal, and I think this is still underestimated. And we have defined about 20 years ago, but there is very old data, I will show it to you, the histopathological growth patterns. And I'm going to take some time to explain to you, based on H&E sections, I'm a pathologist, how these different growth patterns look like. So here you have the what we call the encapsulated growth pattern, was called the desmoplastic growth pattern until a year ago, but the definition has changed, and I will show you why. Where you have liver over here, you have cancer over here. Oh, I have to tell you first, a lot of the data is on colorectal cancer liver metastasis, and the sole reason for that is that not a lot of breast cancer liver metastases are resected. So we have studies, they are smaller, but what I will present you, but there's no reason to assume that the mechanisms will be very different in breast cancer compared to colorectal cancer. but I have to tell you that. So liver over here, colorectal cancer, these are 2 colorectal cancer liver metastases, colorectal cancer over here. And then you can see that in between cancer and liver, there is this scar tissue, which we call a capsule, and hence the name encapsulated growth pattern. And in this scar tissue, you typically have some immune cell infiltrate close to the liver, and you have a proliferation of non-functional bile ducts and also angiogenesis. And I hope you agree with me that this is completely different from this growth pattern, also colorectal cancer metastasis, and this we call the replacement growth pattern, where you have liver up until approximately here, and then you have cancer, and you can see the cancer cells with their pleomorphic nuclei and their increased nucleus cytoplasmic ratio. But it's very difficult for you and for me also to discern the interface between liver and cancer cells. Oh, sorry. And some other examples. Again, the encapsulated growth pattern, cancer over here. You have the scar tissue, the capsule, and then you have the dark blue small cells over here, which are lymphocytes, immune cells, and then you have the liver. And the replacement growth pattern, where you see a linear arrangement of cancer cells that encounter liver cells, and these are the liver cell plates. So in this replacement growth pattern, we have clear cancer cell hepatocyte contact. We also have, and it's maybe difficult to see for you, but we have uptake of these pink cells within the metastasis. So there is an uptake of hepatocytes by the growing metastasis. There is also blood vessel co-option. So this is a sinusoidal blood vessel that starts in the liver and is then co-opted by the cancer cells. So there's no angiogenesis in this growth pattern, at least at the interface. And I think you can also see that the liver architecture is kind of preserved. So what the cancer cell does, and hence the name replacement, it takes the place of the hepatocytes, co-opts the sinusoidal blood vessels, but preserves the liver architecture and makes use of everything the liver has to offer. It's typically an immune desert, and I will not show you the data. We quantified the tumor-infiltrating lymphocytes, and there's a dramatic difference between this density of lymphocytes and the lack of lymphocytes you have over here. And usually these are poorly differentiated tumors, and we like to call this histokinesis. So these tumors do not resemble the primary tumor anymore. They are dedifferentiated, and they have shifted from a primary tumor morphology to a liver metastasis morphology. And then you have the encapsulated growth pattern. I'm sorry, room is too big, too many people. Where you have a fibrous capsule that surrounds the metastasis. There is no contact between hepatocytes and cancer cells. There is angiogenesis in this capsule. The liver architecture, as you can see, is completely disturbed by the metastasis, so not preserved. There is immune exclusion, but you have a lot of immune cells at the interface of liver with the capsule. And usually these tumors are moderately to well differentiated, so they really resemble the primary tumor and the histology of the primary tumor, which is called histostasis. Okay, so now you know the growth patterns. If you look at immunofluorescent images, it's even more spectacular. So this is a replacement growth pattern, and in this case, it's a breast cancer metastasis. And collagen is green, the cancer cells are red, and we did not stain for the hepatocytes. But if you would think away these red cancer cells, it would be enormously difficult to see where the liver ends and where the tumor starts. There is some broadening of the sinusoidal space, but that's it. And this is in contrast to the encapsulated growth pattern. And so we were not the first. This is the oldest evidence of growth patterns. It's in 1888. And this is a primary liver tumor. And this is in French, but what the authors say is that there is very abrupt transition from cancer cells over here to liver cells over there. But they also mention that indeed the sinusoidal blood vessels are co-opted, that there's no fibrosis, and that the architecture of the liver is preserved. And they called it a trabecular growth pattern, and we call it replacement growth. So it's very old, and it's also present, and we also determined this in primary liver tumors. It's also present in primary liver tumors. We wrote with an international team guidelines on how to score these growth patterns in patient samples. So this is the second edition of the guidelines. So in this paper you will find an overview of the studies that have looked at the growth patterns, guidelines for scoring. There is a large multicentric study that clearly demonstrates you the impact on survival of these growth patterns. I will show you the details. And there are some biological hypotheses I will touch upon. And this is really the work, and I have to thank all these people, of the Liver Metastasis Research Network, which is an international group of labs that all do research on liver metastasis and focus on the growth patterns. And we have a meeting each year. If you are interested, you can send me an email and I will invite you. It's a fun meeting. So how do we assess the growth patterns? First of all, you need a resection specimen. Due to the heterogeneity, you won't get there with a biopsy. You need a resection specimen, and then you analyze all the paraffin blocks and all the sections you can get from the metastasis, and you make an average, and that's how you call the growth pattern. So in this instance, and we don't draw the lines, the blue and the red lines, it's just for clarity to explain it to you, but in this, and we just estimate, in this instance, this is a metastasis with a lot of blue interface, which is the encapsulated interface, and then you have some smaller areas of replacement growth. So this would be 70% encapsulated, 30% replacement. There's a high inter-observer agreement after training. We get high kappa values. So this is really trainable, and a lot of people in the world start doing it. And if you then look at the distribution of these growth patterns, when you, and now we have breast cancer data as well, and colorectal cancer data, so every bar is a single patient, and this is the average of all the metastases that were resected, and if it's one metastasis, it's the average of all the slides that were analyzed. And then you can see the typical distribution in colorectal cancer, so about 20% of patients have a pure replacement growth pattern in the metastasis, about 20% have a pure desmoplastic, and then there's a 60% of patients with a mixed lesion. This is different for breast cancer, where you only have a small proportion of patients that have the desmoplastic or encapsulated growth pattern, and the majority has or a mixed or a replacement growth pattern. And there are some hypothetical explanations for this difference. So you see growth patterns independent of the tumor type that goes to the liver. We have demonstrated this in melanoma. Also, you have data in pancreatic cancer. So I think every tumor that goes to the liver will have one of these growth patterns, but the distribution is different. And so what does this mean for a patient? And this you can clearly see from disease-free survival curves and overall survival curves. There's a clear difference in survival between patients that have encapsulated metastasis versus replacement metastasis. And these are large, albeit retrospective studies, but almost 2,000 patients. And every study that is done in colorectal and breast confirms the previous study. So I think it's very solid data. Moreover, if you compare growth pattern with other known prognostic factors, so this is again colorectal cancer, large study, it is an independent predictor of prognosis, independent of, for instance, KRAS and BRAF mutations, or size of the metastasis or number of metastases. So I think that's important as well. And this is the data of one of the papers for breast cancer, and you see the same differences, and it's the same for melanoma, skin and uveal melanoma. It's the same for pancreatic cancer and for other tumors you study. And also in breast cancer, the effect on progression-free survival and overall survival is largely independent of other prognostic factors or other relevant factors for prognosis. Some other clinical features of this aggressive replacement growth pattern as compared to the encapsulated growth pattern. So patients with these metastases have in their metastasis elevated glucose metabolism, as demonstrated by FDG PET. There is a high risk when these metastases are resected on positive margins. Relapse, if it occurs, is more often not limited to the liver, but throughout the body. And patients with replacement liver metastasis more often have circulating tumor cells and tumor cell clusters and elevated ctDNA. And this is also important whenever a new liver metastasis arises during pre-surgery chemotherapy, it's almost exclusively of the replacement type. And another very important observation is, so this is a liver metastasis, it's colorectal, from a patient that got chemotherapy before resection. And then you can see the necrotic center of the metastasis, and you can see the inflammatory infiltrate, and you can see the capsule. But you can also see these mushroom-like outgrowths, and these are of the replacement type. So again, demonstrating to you that replacement is the chemo-resistant phenotype, and we call this escape. and it's very tempting to think of these outgrowths that you can also see in medical imaging as being these replacement outgrowths. So this is, again, what people call the dangerous halo after chemotherapy, again, as a sign of chemo resistance. Okay, so in the last part of the talk, I would like to tell you a little bit of what we think are the biological explanations for both growth patterns. So we've seen they are clearly morphologically different growth patterns. They have a clear impact on disease scores with encapsulated good prognosis or better prognosis and replacement worse prognosis. So there must be a biological difference. And well, it's important that we know this biological difference because we then can probably exploit this to develop new treatment modalities that are really targeting the cancer cell in the new environment, and which is the liver in this case. So we did, this is somewhat older data, we did bulk RNA sequencing, but made sure that we had a targeted sample at the liver tumor interface, so excluding the adjacent liver tissue and not going to the center of the metastasis, so really focusing on the area where the growth patterns are expressed, of course. When you do this and you look at the bulk RNA-seq data, you can see that encapsulated growth is linked to immune response and inflammation, and you see the processes over here. And replacement is very much linked to proliferation and glucose metabolism, so a much more active metastasis, much more proliferating metastasis. This is bulk data. Much to our surprise, we thought growth patterns would link to distinct mutational profiles, and this is not the case. So this is one set of data, and this is together with Memorial Sloan Kettering, 400 patients, and we have a smaller set. So this is DNA-based, and we have a smaller set where we use the bulk RNA-seq data, and we get exactly the same results. So if you look at the driver mutations of colorectal cancer, you don't see any differences if you compare patients with replacement and encapsulated growth. So it's not hardwired by mutations, we think. And then, of course, you have to do spatial omics to get a better view on the biology. And that's what we did with the encapsulated growth pattern, 2 independent studies, and there is a third study that confirms this as well, whether you do in situ sequencing or you do multiplex immunohistochemistry. What you see is that in the liver, you get an acute phase response, so an injury response of the liver. And then, much to our surprise, the capsule is divided in 2 parts, which we call the outer part and the inner part. And the outer part, you get expression of genes that are typically associated with portal tract development and with portal type of fibrosis and with the defense mechanism of the liver. So what we think now is that the capsule is not directed or instructed by the tumor, by the cancer cell, but is an a-specific defense of the liver towards the metastasis. And of course, tumors are very smart, and then they start to modify the fibroblasts, and then you get signals of cancer-associated fibroblasts like FAP-alpha, periostin, PGF receptor beta, and tissue factor. And you see the same over here in the gene expression data. So that would be an explanation or a start of an explanation for the desmoplastic growth pattern. If we look at the replacement growth pattern, it's more difficult. And I want to, I will show you some data, but I want to first get your attention and I want to convince you that there are similarities, purely morphological similarities with liver regeneration. So this is a liver metastasis of a breast carcinoma. Liver over here, a tumor over there. If you compare this image with 2 different animal models of liver regeneration, where you have the new liver cells over here and the sick liver cells in this model over there, and they're replaced by the new liver cells, by the progenitor cells, and the same over here. New liver cells are marked red, the old liver cells are not marked, and they induce apoptosis, so the new liver cells induce apoptosis in the old liver cells. So from a morphological point of view, this really looks like a liver regeneration. And when you look at it with spatial omics, this again is a replacement metastasis, colorectal cancer now, liver over here, tumor over here. And we did CosMx nanostring 1000 gene panel, and asked Cell Charter to define niches or environments that Cell Charter could find. Much to our surprise, you have liver here and you have a tumor over there, but at the interface there are some specialized niches. When you look in these specialized niches, in the dark blue and the light blue or blue-green areas, you can see signs of stemness both in the hepatocytes, the dark blue, and in the tumor cells. So this is what has been called reflected stemness, and we find signals of oncofetal reprogramming. And so what we think is going on here is that you get some kind of competitive liver regeneration. That's why I wanted to show you the images of liver regeneration before. And the same has been described by research from the Crick Institute in London in lung metastasis. And in lung metastasis, you have a similar, we don't call it replacement, but you have normal lung structure. And then this is an immunohistochemical staining where the pneumocytes are stained, or the blood vessels are stained. So you have the alveolar walls. And now there's no air in the alveoli, but there are cancer cells in the alveoli. So again, a cancer cell that is growing in the lung and is respecting the architecture of the lung. Very similar to what is happening in the liver. And also there, you get pneumocytes that go back to a more embryological state, progenitor state, and influence cancer cells to also go back to a progenitor state and to a more primitive epithelial phenotype with fetal reprogramming. So this is probably a more general mechanism of metastasis if they adopt this growth pattern, of course. Okay, and then the next question, we're almost there, is when does a cancer cell, you cannot formulate it like that, but when does a cancer cell decide which growth pattern it will adopt in a target organ in the liver or in the lung? And this is interesting. So we looked at a lot of morphological factors in primary colorectal cancer. And if you do this, you can look at the type of stroma, at the growth pattern, at tumor cell budding, for instance, at tertiary lymphoid structures. If you do this and you combine this information, you can indeed predict what, in this patient, the growth pattern of the liver metastasis will be. So part of the information, and this is interesting for AI, of course, and we're doing this exercise. So part of the information that predicts the growth pattern is already present in the primary tumor. And this is also very important information and exciting information. Some people have identified, by looking at methylation profiles, have identified cancer cells in primary colorectal tumor that already start to behave as hepatocytes and adopt the identity of hepatocytes. And we think if this process is optimized, then a cancer cell will be able to perform a replacement growth in the liver. All hypothetical, and we need to confirm this, of course. So, of course, from biology, we would like to change the disease course for patients, and these are my last slides. So, the aim should really be to develop treatment of liver metastasis that is better adapted to the biology of the metastasis. I think now patients with desmoplastic and replacement growth metastasis are treated in the same way, and probably that's not the correct way to do it. So we have to develop new treatment modalities. We are a bit handicapped in a sense that we need a resection specimen to identify the growth pattern. So it would be much better if we would have markers before surgery and before chemotherapy to identify the growth patterns. And maybe if you think of the methylation profile, ctDNA and methylation profiling of ctDNA would help. And of course, you need to have functional validation of the omics-based data that you have in order to really disrupt the niche of cancer cells in the liver at the advancing front and maybe change replacement to desmoplastic or do other things. And luckily, there is now a good animal model which was absent. So we over the years, we tried to inject, I think, nearly 30 cell lines in spleen or directly into the liver, and you always get replacement growth pattern. Now there's a syngeneic immunocompetent liver metastasis model, and you see the replacement growth pattern over here, very much resembling what happens in a patient, and you have a very nice desmoplastic growth pattern with the immune component as well. So we're writing the manuscript to show to the world that this is a good model if you want to study growth patterns, and if you want to do the genetic disruption of the genes that you think are important. Okay, this is the last slide, so I want to end with, again, a very old observation, 1962. Replacement is the most astonishing and the least comprehensible of the modes of behavior ascribed to metastatic carcinoma. And I think we really have to solve the biology and have to know the biology of this aggressive growth pattern in the liver. Thank you.
All right, thank you. So our last talk before we go into the discussion session is by Rachelle Johnson from Vanderbilt University, and she'll be talking to us about bone metastasis and then biology and treatment implications of that. Thank you.
Great, thank you. So thank you for the introduction and the invitation to speak. It's really my pleasure to round out this panel by speaking on bone metastasis. I have nothing to disclose. So I actually want to start by just talking about the impact of bone metastases on patients with breast cancer. So bone is absolutely a preferential site for breast cancer cells, along with many other tumor types. But when you look in breast cancer in particular, you can see that up to 70 to 80% of patients who succumb to breast cancer actually present with bone metastases upon autopsy. So it's incredibly prevalent, even though it's not always symptomatic. So patients with bone metastases will frequently present to their doctor with fractures or significant bone pain, but they can also develop hypercalcemia as well as spinal cord compression, and typically can end up with really a reduced quality of life. But one of the impacts you'll note that's not on this slide is mortality. So patients with bone metastases are not typically going to succumb to the bone metastasis itself. So, really, why do we continue to really study bone metastases beyond trying to reduce the fractures and the bone pain that those patients experience? And I think there's two really important reasons for this. So the first is that we now understand that breast cancer cells disseminate to the bone much earlier in the disease process than we originally thought. So we thought that if we caught the breast tumors early enough, that we would prevent dissemination into the bone. That does not happen. So there's really good evidence from Christoph Klein's group from about 15 years ago that showed that in mouse models of ductal hyperplasia, so precancerous, and in patients who have ductal carcinoma in situ or stage 1 breast cancer, you can actually find cytokeratin positive, what will ultimately become breast cancer cells in the bone marrow in those patients and in the mouse models. The second reason that I think this is really important is that we now understand that breast cancer cells that disseminate into the bone marrow actually seed the soft tissue sites to form soft tissue metastases. And so the earlier and better we can do in preventing bone metastatic progression and trying to target cancer cells in the bone, that will contribute to the overall mortality and prevent some of those secondary soft tissue metastases. So I just want to spend a little time talking about the biology of breast cancer cells and how they get to the bone and engraft in the bone. So breast cancer cells are attracted to the bone by ligands that are secreted both in the endothelial cells as well as by osteoblasts that line the bone surface. So a good example of this is CXCL12 that's produced by the endothelial cells and osteoblasts, and this binds to the receptor CXCR4 in breast cancer cells. So there's an actual pull factor and a gradient that brings the breast cancer cells into the bone marrow. Once the breast cancer cells get to the bone, they will encounter this normal bone homeostasis cycle, which is where you have balanced bone resorption and bone formation. So osteoclasts are the bone resorbing cells. They will be recruited to sites of microdamage or old or damaged bone, where they'll resorb away that bone, and then they will secrete factors that recruit osteoblast precursors. The osteoblast will then form new bone in its place. So this is a really tightly coupled process. It's tightly regulated over your body, and it's happening in everyone right now, all over your skeleton, and you actually get roughly a new skeleton about every 10 years due to this process. So when breast cancer cells come into this environment, they completely disrupt that bone homeostasis and the balanced turnover that occurs. So breast cancer cells secrete factors that really promote osteoclast resorption. So a key factor in this process is parathyroid hormone-related protein, or PTHrP, which will bind to the osteoblasts on the bone surface and cause secretion of Rank ligand, and this ultimately fuels osteoclast differentiation. When the osteoclasts start to resorb the bone, they release all of these growth factors and cytokines that are actually stored inside the bone matrix. So the bone matrix is a reservoir for these. And this gets released into the tumor bone microenvironment. It acts back on breast cancer cells. It fuels their growth and proliferation, and it also further increases secretion of those factors like PTHRP. This can be called the vicious cycle of bone destruction. It's also called tumor-induced bone disease. It has several names, but the effect is the same, that it really, breast cancer cells have a way they can fuel their own growth in the bone. So I wanna focus a little bit on PTHRP because it's a really critical step in this cycle. So really our understanding of how PTHRP drives bone metastasis comes from two studies that were in the 90s. So the first is that a group took breast cancer cells, these were MCF7 human breast cancer cells that are not very bone destructive, and they overexpressed PTHRP in those cells. And upon doing so, you can appreciate the cells became very lytic, bone destructive, and they grew out really well in the bone. Conversely, another group used PTHRP neutralizing antibodies and showed that if you take breast cancer cells that do tend to cause osteolytic bone destruction, the MDA MB231 human breast cancer cells, when you use the PTHRP neutralizing antibodies, you block that effect. So this really led to several more decades of research into PTHRP, and so that we could understand its biology and if we could target it. And my lab has been one of those groups that's been working on it. And we've found that, like everything else in science, it's far more complex than we originally thought. So PTHRP, this is just a schematic of the molecule here, but it is actually proteolytically cleaved all over the place. There are well over a dozen sites at which this molecule can get chopped up. And so what my group has done is we have created these different mutant versions of PTHRP where we've deleted different parts of the domain. And this is just a few of them, but we have kind of a whole suite of these now. So what we found is that depending on which part of the protein you delete, you get really different and dramatic effects on cancer progression. So this is actually in the primary breast tumor site in a preclinical model. We find that if you delete just the nuclear localization signal in PTHRP, you actually speed up tumor growth and you get much larger breast tumors. But if you delete the entire C-terminal portion of the protein, you actually get the opposite. You slow down tumor progression and you get really small tumors. So all we did was just change this small part of the protein that was expressed or not. So really this, I think, I just want to highlight this because I think it really nicely portrays how we had this target like 20 years ago that everyone was excited about, and then when we really started to dig in and think about it, it became far more complicated. So there are groups now that are looking upstream and downstream of PTHRP, and we're looking downstream of these different fragments now to see if there's better targets. So when we think about where breast cancer cells like to engraft within the bone, there's a few different niches where they can go. So the first is the perivascular niche. So this is truly just along the vasculature. And then the second and third are the hematopoietic stem cell niche and osteogenic niche, which actually overlap. So the osteogenic niche truly just means the surface of the bone. It can also be called the endosteal niche. And the HSC niche sits right next to those osteoblasts. So HSCs actually like to sit next to the osteoblasts where they'll get these prodormancy cues from the osteoblasts, and that prevents them from proliferating. So it preserves them in a more dormant state, which is really what you would want for your stem cells. So there's actually not great evidence for breast cancers displacing HSCs. Those studies have not been done. There's good evidence in prostate cancer, but we do know quite a bit about the perivascular and osteogenic niche from studies just in the last 10 or 15 years, and especially in the last few years. So in the perivascular niche, we actually now know that cancer cells, depending on where they engraft along the vasculature, will either remain dormant or start proliferating. So cancer cells that engraft along the quiescent stalk of vasculature, where it's very stable, those cells remain dormant, whereas breast cancer cells that that end up next to sprouting vasculature, those cells actually start to proliferate. So the spatial orientation of cancer cells along the vasculature is incredibly important for whether or not they'll progress in the bone. When we look at the osteogenic or endosteal niche, we now know that in addition to the breast cancer cells stimulating osteoclastogenesis, like I showed you through PTHRP, the breast cancer cells also secrete factors that are chemoattractants to bring in even more osteoclast progenitors into that site. We also now appreciate that breast cancer cells interact directly with osteoblasts as well. So there are now osteoblast tumor cadherin interactions that have been identified that regulate breast cancer progression. And we now understand that the calcium that is being secreted by the osteoblasts and coming out of the bone can also fuel breast cancer growth in the bone as well. So we've learned quite a bit over the last few years even about how cancer cells interact with cells in the bone. And this is actually pretty impressive because we do not have very good preclinical models for bone metastasis. So I want to take just a moment to talk about how we model and how we detect breast cancer cells in the bone. So these are some of the cell lines that the research community has relied on heavily for bone metastasis models. You have human MDA and B231 cells, human MCF7 and T47D cells, and then mouse 4T1 cells. So all of these have pros and cons. These cells are all human, which is good, but it's bad because they all have to go into immune-compromised mice. The 4T1 cells are able to go into immune-competent BALB/c mice, but they are so aggressive. It makes it extremely difficult to look at them in bone metastases because they just take over the bone marrow so quickly once they get in vivo. So they can, they're good for really aggressive disease, but really challenging at times to work with. So the common injection routes that we use for these cell lines is that we will typically introduce, the most common is to introduce these cells into the bloodstream. So you can do this by intracardiac injection or intrailiac injection. So this is intracardiac goes into the left cardiac ventricle, intrailiac goes into the iliac artery, and these are both models that are truly models of bone colonization. So they're not a true metastasis model, they skip the intravasation step, but they model the rest of the metastatic cascade pretty well, and both of these are effective ways to get cells into the bone. The other options are intratibial or intrafemoral, where you directly inject the breast cancer cells into the bone itself. This is a really good way to look at tumor growth in the bone. It is definitely not a bone metastasis model, but it can be really useful if you need to know where the tumor cells are going to end up in the bone, like if you're doing an imaging study. There's also the option for orthotopic models of bone metastases, and this is probably the least commonly used, even though it's what we want to use the most. So, and the reason why we don't is because there are no reliable models for orthotopic bone metastasis. 4T1 is the closest we can get to that, but the mice will typically succumb to their lung metastases before they can really develop advanced bone metastases. So, our lab in particular has been focused on ER-positive bone metastasis models, which are the hardest to work with. So we've used MCF7 and T47D because there are no ER-positive, no reliable ER-positive mouse models for bone metastasis. And so these are the ones that we focused on, particularly MCF7 cells. But what we have found is that these can be incredibly difficult to find in the bone. So this is some work we did several years ago where I just want to draw your attention to the top here. So these are mice that we injected with MCF7 cells by intracardiac inoculation. We did not give the mice estradiol here. And when we looked by histology, there's multiple ways you can look for these. You can do histology, you can do bioluminescence imaging, you can use a fluorophore, different optical imaging modalities, but we used histology, and we could not detect these breast cancer cells by histology. When we added estradiol, then you can see that you do start to see some of these breast cancer cells growing out in the bone. It's not massive metastases that are forming, but we can absolutely start to detect them there. But what you can also appreciate when you look over here is that when you add estradiol, in addition to all those effects it's having on the astrocytes and in the brain that we learned about, it is dramatically altering the bone. So this is what the bone normally looks like. You have a growth plate. The orange or pink is the bone. This is cortical bone, and there's little bits of trabecular bone in here that are also orange and pink. The purple is your normal marrow. Here you can see there is practically no marrow left at all. It is almost entirely bone and mineralized bone. So you've completely disrupted the niche. So what we wanted to know is, could we find ways to better detect the tumor cells in this model without estradiol, and then potentially use this as a model where we could, if we wanted to do proof of concept, to try and push the cells sort of out of a more dormant state. So we tried several different modalities. We ultimately ended up on using flow, so we'll actually flush out the marrow out of the bones, do flow sorting after we have stained for CD298. So CD298 is a great marker for human cells, and it works really well to find human breast cancer cells in the bone marrow. So it stains absolutely nothing in the bone marrow in the absence of breast cancer cells, but then when we added MCF7s, I don't know if you can see it on that screen. You can. There are 2 cells here. This is shockingly reliable. So you will find a few cells on the background of no cells in the absence of cancer in all of the mice when you stain for this. And then when we add estradiol, we enrich for those same cells. So we feel good about we're going after the right population. So I have added this as well. We have tried these different modalities on different breast cancer cell lines to see if we can find some other alternative models, and I've published all of this, so I'm just putting it up for if anyone is interested in looking at bone metastases in their models. These are some potentially good resources for you here in this paper. Okay, so because we have relied so heavily on these human breast cancer cell lines, we obviously know really not a whole lot on how the immune system is impacting bone metastases. So our group and others have recently shifted towards the E0771 breast cancer cell line to try and investigate this. These are ER negative. This is some imaging of the pan-CK is for the E0771 cells. So you can see we have green tumor that's growing out in the bone here. And then CD45 is just a stain for all the immune cells, and cyto13s are nuclear stain. So what I hope you can appreciate is that there are a few pink cells that are scattered in here in the bone and in the tumor, but you can really nicely see how immune excluded the bone is. So right here along this margin, this is where the cortical bone was and is almost gone because it's been destroyed by the tumor cells, but just outside of it is all these CD45 immune cells. So this was really fascinating to us. We saw this across multiple mice when we did these stains. And what we are now doing is focusing on, we're using digital spatial profiling to see, can we look at what immune cells and what factors are enriched in the immune cells that are close to that endosteal or osteogenic niche versus the immune cells that are out here in the marrow that are not right next to the bone. So this dark piece is actually a piece of bone. So just looking qualitatively, this is a myeloid panel. You can see that the immune cells next to the endosteal surface in tumor look different from the immune cells that are hanging out in the marrow next to the tumor. And some of the factors we see enriched in the endosteal niche is actually PD-L1, PD-1, myeloid cells. So all cells and factors that could potentially influence tumor killing as well as tumor progression and certainly immune interactions with the tumor cells. So this is something we're following up on. Okay, so this brings me to the current clinical treatments for breast cancer bone metastasis. So right now, there are 2 different ways that we can treat bone metastases, and they both target osteoclasts. So the first is bisphosphonates. These are drugs like zoledronate and alendronate, and denosumab, which is an anti-RINC ligand antibody. It's called Prolia or Xgeva. And these both target osteoclasts, but they do it in slightly different ways. So the bisphosphonates actually kill the mature osteoclasts, so they induce osteoclast apoptosis. But denosumab actually prevents osteoclast formation. So it's this RINC ligand antibody, so it will block this progression from pre-osteoclast into a mature osteoclast. So you can still get accumulation of osteoclast precursors, but they do not fuse and become a mature osteoclast. So just 2 slightly different mechanisms. But they're both anti-resorptive, so they block bone resorption. So there have been multiple clinical trials over the years that have tested out zoledronate versus placebo, denosumab versus placebo, and then comparing zoledronate to denosumab. So I just want to highlight a couple of the studies here, and they are not without controversy. So the first study I want to talk about is the AZURE study. This found that zoledronate versus placebo increased overall survival in postmenopausal women specifically. So it was very effective at increasing survival. However, in premenopausal women, it actually made things significantly worse. So, and this was because zoledronate actually increased extraskeletal metastases or soft tissue metastases in patients who were premenopausal. So it is now indicated for patients who are postmenopausal, where it's very effective at blocking bone metastases and does not have those extraskeletal metastasis issues. The next study to highlight is this SABCS 18 which found that denosumab versus placebo reduced the risk of fracture and increased bone at all sites in patients with breast cancer. And in the follow-up, it showed that there was a small but significant increase in bone metastasis-free survival. So this is all very promising outcomes for patients. However, at the same time, there was a second study done that was called the DCARE study, and this was also with denosumab versus placebo, but in a slightly different patient population. And so with this study and the DCARE study, again, this was denosumab versus placebo, they found that there was no effect of denosumab on bone metastasis-free survival. So it just muddied the waters a little bit, made it a little more murky about the effects of denosumab on bone metastasis-free survival. But certainly denosumab is still effective at reducing fractures and increasing BMD no matter what. So denosumab was also compared to zoledronate in the study here. So denosumab actually reduced skeletal-related events more effectively and increased quality of life compared to zoledronate in the study, but the overall survival was the same between patients who were taking zoledronate or denosumab. So collectively, what this data really, where it sort of lands right now is that the recommendation is to either give denosumab or zoledronate, but that's pretty much it for the guidance. So in the research community and in the bone field, there has been additional interest in denosumab and how it's working, especially in the context of breast cancer bone metastasis, because it has this rebound effect that's very well documented upon treatment cessation. So when you end denosumab treatment, for whatever reason that may be, that the patients, because you have blocked the fusion of all of those osteoclast precursors, but they're still there, as soon as you take denosumab away, all of those precursors can now fuse into osteoclasts. And so what you get is this bolus of bone resorption. You get this spike in bone resorption markers in the serum, and you get this loss of bone that occurs that you can see here. This is after, so this is where you end treatment, and then you get this dip in bone, but then it levels back out. So it's just a window of time where it's like the bone has to recover after. So the concern has been, will those patients fracture? And will that be a problem for patients who have breast cancer or breast cancer bone metastases? So in what's been reported so far is that there's one study that reported in 15 patients, there were 60 fractures that were reported in those patients with breast cancer. And this was in the 2 years after discontinuation of denosumab. There's also been high rates of recurrence after denosumab withdrawal in several studies of patients with giant cell tumor of bone. And again, so our major concern from sort of the research and preclinical standpoint when we're thinking about this is we know that fractures stimulate growth of bone-disseminated breast cancer cells. And so, you know, we would just want to be mindful of that. And I think all of us agree that we just want more preclinical studies to figure out how breast cancer cells and osteoclasts are interacting in the context of denosumab. So this brings me to my last section, which is thinking about other potential therapies going forward. Are there other ways that we can target the bone and prevent fractures and improve outcomes in patients with bone metastases? And so one of the options that we have recently started to look at is bone anabolic therapies. So the two I'm going to focus on today are teriparatide and abaloparatide. There's a third one called romosozumab. But these drugs are all FDA approved for osteoporosis, and they work by increasing bone mass. So where the anti-resorptives block further bone resorption, they do not restore the bone that was lost. The bone anabolic therapies do that. So they actually actively increase bone formation, so they can restore bone. The concern has been that they had a black box warning about concern for osteosarcoma until just a few years ago. And that was because in the original studies for teriparatide, they found that rats who were given teriparatide for 2 years daily developed osteosarcoma. And so that was very carefully monitored when they rolled out teriparatide, and they found that in about the 20 years since it's come out, that does not show up in the real world. So there's no increased incidence of osteosarcoma or any other cancers in patients. So they lifted that black box warning pretty recently, but there's still a caution statement that's on the insert in that box that really warns and cautions against use in patients with bone metastases or a history of skeletal malignancies. And I don't have time to go into the biology of this, but part of that is because teriparatide is a PTH analog and abaloparatide is a PTHRP analog, which, as I showed you at the beginning, can fuel bone metastatic progression. And so that was really the big concern there. We've done a lot of preclinical studies that show that PTHRP actually doesn't act back on the primary breast cancer cells, and so I can, if anyone has questions about the biology of it, I'm happy to answer those in the Q&A. But we now have some of the first preclinical data that I'm showing you for abaloparatide effects on breast cancer bone metastasis and the MDAMB231 model. So we injected those cells by intracardiac injection, gave daily treatment with abaloparatide for 24 days, and then we looked at the bone and the cancer in the bone. So when you look at the bone first, I think everyone can appreciate this is a massive increase in bone. So this is what we see when we treat with abaloparatide. So we can absolutely still increase bone formation in this model. And when we looked at the cancer cells using our CD298 flow marker, you can see that there is no change in the cancer burden in the bone. So this was actually a positive outcome for us because we just wanted to make sure there wouldn't be more cancer there. And there's not, it's the same. So this was exciting. It suggests maybe this could be paired with other adjuvant therapies or even with anti-resorptives as well to potentially improve outcomes for bone. So just very briefly, the key take-home points, I think, are that breast cancer cells go to the bone, again, much earlier than we thought, and they seed those soft tissue sites. That they also promote bone breakdown by secreting osteolytic factors. They tend to graft along the vasculature and in that osteogenic or endosteal niche, that they frequently do disseminate to the bone, but they can be really difficult to detect. And probably many of the cancer models that people are using probably actually have cancer cells that disseminate to bone, but you have to look really hard for them. Anti-resorptives are currently our best way to treat bone metastases, but we're looking at some other options and other bone-targeted agents to see if they may also help improve outcomes. So with that, I just want to thank the lab, acknowledge the Cancer and Bone Society as well for those who are interested in learning more about bone metastasis, and our collaborators in funding. Thank you.
Great, thank you to all of our speakers. So before we open the floor up to questions, we wanted Christine Hodgdon, who again is an advocate who works with the Sidney Kimmel Cancer Center at Hopkins, has been involved in a lot of advocate programs like GRASP and Metastatic Breast Cancer Alliance, specifically with respect to her interest in brain metastases, is going to talk to us a little bit about her thoughts around metastatic disease, and then we'll open it up to questions.
Thank you, and thank you for inviting me for this panel. My name is Christine Hodgdon, and I've been living with metastatic breast cancer since 2015, so just close to 10 years. I co-founded an organization called GRASP, as we were mentioned, which seeks to bring patients and researchers together for meaningful collaboration. I had metastasis in the one organ not discussed today, in the lung, but over the last decade I've learned a lot about metastasis, and because I'm an overachiever, I met with each speaker individually to better understand the research presented, so I'd like to summarize in patient-friendly terms what I learned and where this research will have the greatest impact. So in addition to GRASP, I do lead a brain metastasis initiative called the Marina Kaplan Project, composed mostly of patients actually living with brain metastasis, and the majority with ER-positive breast cancer. And they always ask this question, why don't I ever see any brain mets research for my subtype? It's always HER2 positive and TNBC. So this became an area of research we identified as lacking. And in walks Diana with her research. So I didn't have to meet with Diana because I've been collaborating with her for years. And her research elucidates a novel mechanism for progression in the brain whereby estrogen suppresses the brain's immune defenses. I believe that Diana's collaboration with these brain mets patients helped highlight the far-reaching impact her work would have in the management of brain metastasis for breast cancer patients. Yesterday, Peter kindly met with me to discuss his research in liver metastasis and explained to me this idea that not all liver mets are equal, that in fact, two unique growth patterns exist, which may inform how the metastases are treated. Liver metastasis is a big problem for patients. 5-year survival rate is less than 10%, which is significantly lower when compared to metastasis in other organs. I understand it is not standard of care to do liver resections in breast cancer the way it is in colorectal cancer, which is why we saw some CRC data in your presentation. But might we be able to discover a surrogate, like a biomarker, something less invasive that can help identify different growth patterns? I also happen to know plenty of patients that would happily undergo a liver resection if it meant gaining insights into a treatment personalized to their specific cancer. I always like to remind researchers that patients are actually your biggest supporters. We are willing to do a lot because our lives depend on your success. I next spoke with Rachelle, and I learned a lot about bone metastasis. And I learned that when breast cancer spreads to the bones, we often see that it takes root in the blood vessels and can seed or travel to other parts of the body, like the liver and lungs. Again, I had never heard of this. And she even described a really cool study that we don't have time for that proved this, but if you want to talk to us after, we can tell you about it. So while the 5-year survival rate is higher for patients with bone metastasis, we also know that both the metastasis and the treatments can cause debilitating chronic pain and fractures that significantly impact quality of life. Another important point for patients with bone mets only, they're often excluded from clinical trials, so we really need to do better for our bone mets patients. There appears to be some conflicting studies on bisphosphonates versus denosumab, but what I hear from patients is that we need more research to flesh out what treatments are going to prevent or stop the metastasis while simultaneously preserving quality of life. We likely need to better educate those healthcare providers treating the side effects of these drugs, like osteonecrosis of the jaw as well. It also appears that, like brain metastasis, we need better animal models that can replicate metastatic progression as we see it in a patient with ER-positive disease. So what else can I say about metastasis other than it's complicated, not only for patients living with metastasis, but it's complicated for clinicians treating these patients. It's complicated for the researchers studying metastasis. But it is precisely because of this complexity that I'm so grateful to the clinicians and researchers who dedicate their careers to studying these really difficult and nuanced challenges. And the message I want to send to the researchers in the room today is to remind you that you do not need to tackle these challenges alone. There are throngs of patients and advocates ready and willing to collaborate and to help highlight the true impact of your work, or tell you when the work won't be that impactful, but to also amplify your findings. So thank you.
Great. So we have a number of questions in the chat. I don't know if anyone in the audience has questions. If so, if you can just come up to the microphones. It's a bit hard for us to see because the lights are glaring in our eyes, so you might need to scream out a little bit just to let us know if you're there. In the meantime, I can get started with some of these questions. One of the first questions, Deanna, that came up was for you, and it was around, obviously, brain metastasis. And the question is, if the use of estrogen therapy is the reason for fewer brain mets in ER-positive breast cancer patients, why do we observe more brain mets in the triple-negative patients in de novo metastatic breast cancer than in hormone receptor positive?
Sorry, I missed the beginning of your question. Can you read it again?
Because you said that estrogen therapies or estrogen inhibitory therapies, right, are the reason for fewer brain mets in ER-positive breast cancer patients. So they're wondering why there are more brain mets in triple-negative patients, de novo, who don't have estrogen.
So I think I'm not sure I understood completely the question, but there are more. So one of the reasons there might be more triple-negative breast cancer brain metastasis de novo is in part because most women are pretty young by the time they have the primary tumor. And as we learned from Rachel, the dissemination of cancer cells, unfortunately, more and more studies have demonstrated that this dissemination is something that is happening much, much earlier than even the primary diagnosis of tumors. So most women and patients with breast cancer might already have disseminated cells to different organs, and whether those disseminated cells are able to grow in those distant sites is a function of the intrinsic aggressivity of those tumors, which triple-negative tend to be very, very aggressive, and also that microenvironment. And so what we think is that, high levels of estrogen in premenopausal women, most of them with triple-negative breast cancer, might be contributing to that early onset of the raise of dormancy from these disseminated cancer cells, while patients with estrogen receptor positive breast cancer that have disseminated cancer cells but are receiving or will receive endocrine therapies very early on might find a delay sort of in that raise of exit of dormancy is our hypothesis that is partially contributed by endocrine therapies. Like I said there, the caveat with this is that we know endocrine therapies are also targeting the tumor. So it's very difficult to really dissect whether that is truly happening.
Great, thank you. Looks like there's a question at the microphone there. We can take that.
I'm Luciana from Texas Tech University. My question, first of all, thank you everybody for amazing presentations. My question is for Dr. Johnson about the abaloperatidine. Have you ever tried, the dose that you're using, is it the current dose for the patients? And have you ever tried the same approach for different models, such as canine models? And if you have observed, what will be the major side effects that you observed?
Yeah, that's a great question. So, for the abaloperitide, the dose that we use is 80 micrograms per kg. And we really struggled to figure out which dose to use for that. What we ended up doing is that is truly a maxed out dose. It is higher than what you would give a patient. And the reason why we did that was because we were mostly concerned in that study about, can we potentially stimulate cancer cell proliferation? So we wanted to hit it as hard as we possibly could with that drug to figure out, do we see something? And then we were going to titrate back from there. So again, that was our first study. We are planning on titrating it down. So what would be closer to patients would probably be more around like the 20 to 40 micrograms per kilogram dose. And so we are absolutely planning to do that. We have not tried it in canine models or in other models as well. Again, this is very new data that we're collecting. But yeah, we would absolutely love to move into other models. So in terms of side effects that you mentioned, we haven't, so I'll say on the preclinical side, we have not observed any in the mice. We have also looked at the immune compartment. It looks relatively normal. There's some changes in the myeloid cells, but there's not anything dramatic in terms of like neutropenia or anything, so.
Thank you. We have another question?
Yeah, hi. Oh, sorry. Thank you so much. This has been really, really interesting. My name is Jade Moran. I'm a patient advocate. I'm a research advocate. And I've been living with de novo metastatic invasive lobular carcinoma with bone-only mets since 2022, so 2 years. And I do take Zometa, so I'm familiar with this whole spiel. I have a question about ILC versus IDC, but I have my main question is about exercise, or rather bone loading, okay? So the first question is, if there is no difference, that's fine, but I'm curious about how breast cancer subtype is relating to all of these things, or if it's rather just about the sclerotic versus lytic, because I know that in ILC, it's often a diffuse, innumerable mets that are kind of a little bit less lytic in nature. At least mine are sclerotic only. So that's one question. And the main question that I have is, I read a very, very interesting study out of Cornell, obviously in mice, but so we can't extrapolate too much, that showed that bone loading made for an 80% reduction in the spread of metastatic in the bone for, moderate. There's a J-shaped curve, actually. If you go too much, much worse for you. So first, recognizing that it's not always possible for people with bone mets because of pain and other mobility issues to exercise. And also, I wanted you to comment on that. And also, I know that vibration is often used for elderly patients who are bedridden to help them maintain their bone density. So I was curious about that in relationship to people with metastatic bone-only or bone disease. And finally, I know this is a long one, so you can take whatever part of it you need. Now, this is actually one that I really cannot find any literature on. If a patient does do exercise, bone loading, in order to improve their bone density and reduce the spread of their disease, does it matter does it affect the local area only, the bone that is loaded only, or is it a more global effect? So, sorry, I just unloaded all those questions on you, and thank you so much for all of your presentations.
Yeah, no problem. Those are all fantastic questions. So I'll start with the muscle and exercise. So honestly, it has not been well studied, but there are studies that are starting to come out on this. And there's some research in particular from Tara Ballinger, who is actively working on this. She is coming up with clinical trials of using loading exercises to really see if it does improve outcomes. But we just don't have the data yet, so that's still early on, but it is actively in the works. So I would stay tuned for that, because that's obviously a really important question. In terms of the mouse study and loading on the bone, so loading on the bone it increases bone formation. That's typically how it works. And so you're right, there are some preclinical studies that look really promising for it. When we look in patients, so for example, with like vibration and using something like that to increase loading, it doesn't always translate to the same impact in patients as what we see in a really highly controlled mouse study. And there's probably a lot of reasons for that. We're bipedal and mice are not. So there's a lot about loading studies that don't always translate, which can be frustrating. So I think right now, all of that is going to really play out in those clinical studies where they're looking at the different types of exercise. So I think we will hopefully have that information soon for you. And then I think you mentioned lastly about if a patient does exercise, is that affecting the local area or is it affecting it globally? So it's very likely affecting it globally. And that's because the bone is an endocrine organ. So when you start to impact, and when you start to load one particular bone, you're going to change what's being secreted and what's being released from that bone, and that's going to affect the entire body. So, it can absolutely have a systemic effect.
Thank you.
Thank you. So I'm going to ask a liver met related question. So with respect to the different types, the replacement type versus the encapsulated type, you mentioned, I think, and I may have gotten this wrong, that you can actually get information from the primary tumor, the histology of the primary tumor, to predict, right, whether it's going to be more replacement or encapsulated. And I'm trying to put that data together with the data that I think you showed that from a genomic standpoint, these two different types don't look any different. Mm-hmm. So I would have, I guess, from those data together, thought that it had something to do with plasticity and migration and interaction with the tumor microenvironment. So I'm surprised that you can predict that from how it looks in the primary tumor. And I guess I wanted to know your thoughts about that. Why do you think that that's predictive, and what are we missing? It's obviously not genomic, so there's got to be something else going on in the primary tumor already that's dictating whether you get the replacement or the encapsulated forms.
Yeah, that's of course a very important question, and we don't know yet. But if you look at melanoma studies, there you, for instance, see cancer cells that are lying around the blood vessels and further away, and the cancer cells around the blood vessels are the ones that disseminate and have a stem cell phenotype, and the other ones are responsible for the growth of the primary tumor. So maybe something is happening here as well, where you have in the more aggressive de-differentiated primary colorectal cancer, and I think the same will apply to breast cancer. You have other niches than in other tumors that are more differentiated. So I think I think what we see on H&E are a reflection of these different niches, and that if we understand this better, and if we maybe get attention maps from AI, where AI sees the differences, we will be able to define these niches and then find the cells that start to behave as hepatocytes already in the primary tumor that then will give rise to preferentially the replacement growth pattern. But that's still a wild guess.
Thank you. Looks like we have another question from the audience.
Yes, thank you very much for the great talks. Dr. Gaia Griguolo from the University of Padova. I have a question for Dr. Cittelly on brain mets. I was extremely intrigued regarding the interaction between FGFR1 and the different aging of astrocytes. So I wanted to just ask you if do you expect that according to your preclinical data, we should find different levels of genetic alteration of GFR1 according to the age of the patient? Because that would be something really interesting to test in clinical courts, just to understand if this is relevant in patients and then go on to test this from a therapeutical point of view. Thank you very much.
Yes, no, that's a great question. So I don't think our data suggests that there is an age dependency on the acquisition of FGFR1 mutation. I mean, I think what we have learned is that FGFR1 amplification is in part a result of endocrine therapy resistance acquired by the tumor cells, or potentially one of those genetic aberrations that are part of the drivers of tumors at primary sites. What might be changing, it's the ability of those cells that have acquired FGFR1 amplification to thrive in different organs, and I was specific to the brain, that have the molecules that are able to activate the pathway when the amplification itself is not the driver. It's not an amplification that is causing the signaling to be amplified themselves. And so what we think and our data suggests is that in younger females and in younger patients that have high levels of estrogen and high levels of FGF2 in the brain, that tyrosine kinase activation of the pathway in FGFR1-driven growth is fueled, if you want, by that normal high level of FGF in the brain. And that particular pathway is likely to be decreased in older women, which could mean that if the FGFR1 inhibitors that are available only target that kinase activity, they might be more effective in younger women, but not as effective in older ones. With that being said, NCAM so far seems to be able to induce a similar kinase activity, which would suggest that in that context, the inhibitor might be equally effective in both older and younger women, just activating different or targeting different roles, if you want, of FGFR1 in those microenvironments.
Thank you. I'm going to read another question that came up in the chat, and this is for Rachelle. What do you think about local treatments in bone metastasis like cryoablation, electroporation, SRBT, and surgery?
Yeah, so I think it really just depends on really what the patient's disease looks like. So those are really typically only going to be options for patients who have like one bone metastasis or who have, just a few lesions in one place in the skeleton. And so I think in that case, I say this as a non-clinician, that it certainly, that those can be effective therapies. So I think when we talk about treating bone metastases with like the anti-resorptives or the bone anabolic therapies, we're thinking about that in the context of patients who have systemic bone metastases, who have more than one and who have them all over the skeleton. So, yeah.
Great, thank you. Another question from the chat. This one is for Peter. If liver mets are preferentially infiltrating bile ducts, does it belong to the replacement type or is it a separate entity?
Oh, wow. I think you see that combined with the two growth patterns and mainly with replacement, of course, the intraductal growth, which then seems to, well, this is another hypothesis, seems to resemble a backfall to in situ growth again, because then they, and in this case, they also replace the cholangiocytes that are lining the bile duct, of course, but respect the basement membrane. So this is a kind of reversal towards in situ growth. But it's not a separate growth pattern. It's linked to mostly replacement. And what you, well, in association with that, there are several other growth patterns that are not that frequent, and I didn't discuss it. You have pushing where you don't have the desmoplastic rim or the fibrous rim, but you also have no infiltration in the liver. And then there's a very interesting growth pattern where the cancer seems to spread in the liver via the portal tracts with an affinity to the portal stroma. And there you also see the intraductal growth, the bile duct growth.
Yes, of course.
So I think just following up on that, and I don't know if I missed this from your talk, but is there any relationship between the patterns, those two different patterns that you have, and different breast cancer subtypes? Can you see whether, like, ER ones are more, versus the other?
Yeah, so not at this moment, but I think the cohorts were too small to really establish that. And in colorectal cancer, we don't see any association with CMS subtypes or the more modern ways of categorizing colorectal cancer. So I presume it will also not be the case in breast cancer, but I don't know. It's too early.
So another question, there were some immune questions with respect to brain metastasis, and I'm going to sort of layer on top of that because there were people who asked about whether the immune microenvironment or the lack thereof favors metastatic breast cancer to the brain. But my question for you, in addition, is do you know which immune cells are actually cooperating with the radiation therapy? So what is that innate? Is it adaptive? Is it a combination that's actually making that so effective?
Okay, the short answer to that second part is we don't know fully yet. We suspect that is a subset of myeloid cells that are the ones responsible for that, and dendritic cells. But we don't know fully that part of the story. Also, mostly because when we arrived to these findings, we really kind of arrived by them by chance. We were looking at sort of the role of endocrine therapy in this therapeutic setting, and we were quite surprised when the therapies will only work in the context of radiation, but not alone. When the immune changes in microglia and the recruitment of T cells, B cells, and NK to the brain, they are happening regardless of whether the radiation is there or not. So there is clearly a component of immune suppression that we think is partially due to myeloid cells and T cells that is somehow bypassed when there is radiation. And so what we think is the radiation-induced antigen presentation might be a key component to the effectivity of that. And sorry, I forgot the first part now.
That was a question in the chat, the first part, which was whether there's a lack of TILs in the brain to begin with, and things that might favor the growth of triple-negative breast cancer.
Yes. So I think when one thing that is sort of different from the brain from other organs is that there is really no T cell and all of these immune cells through infiltration into the brain parenchyma unless there is a injury or a trauma, and in this case, brain tumors. And all of the studies that I show, it really require us to look at a pool of immune cells in a full brain, not unlike primary tumors in which you can look at the lymphoid cells associated with the tumor. Specifically in brain metastasis, at least in the mouse model, we cannot truly isolate little mets to do that analysis because the numbers are just too small. So what we see, though, is that, and in the brain this is important, infiltration to the brain of T cells is very important, because the majority of brain metastases really don't grow as big balls of tumor, but in the brain they tend to grow around the vessels. So they are sort of co-opting vessels and hugging the vessels as they grow. And so many of these cells are actually much closer to the brain metastasis than they are in a very deep, solid tumor. But with that disclaimer, we have not looked at that digital pathology molecular level to see where are the cells. And I am absolutely sure there is going to be differences in the patterns of where the cells are arriving in the brain, but we haven't done those studies yet.
Great, thank you. I apologize that I haven't gotten through all the questions in the chat, but I think we were told we had to stop right on time, so I think we should thank all the members of the panel today for their great talks and contributions, and move on to the next set of talks here. Thank you.